Greetings. Let's continue with the arterial blood gas. So, I think last time we talked about some basics of pH, and then we looked at uh, some variables. We looked at pH, which gives you acidemia or alkalosis, and that's body's overall ability to donate protons or accept protons. We had partial pressure of carbon dioxide, which was the respiratory, and it can give you acidosis and alkalosis because that's a process. We had bicarb, which was the metabolic side that can give you acidosis or alkalosis depending on what's contributing. Then we have oxygen, which partial pressure of oxygen, which is the oxygen dissolved in the blood. It gives you whether a patient is hypoxemic or not, right? What was normal pH? 7.35 to 7.45. What's PaCO2? 35 to 45 millimeters of mercury. What's bicarb? 22 to 26 milliequivalents per liter. And what's PaO2? 80 to 100 millimeters of mercury. Okay, do I have to remember them, these values? Yes, you do. Sorry, but we need to know these values. Normal pH, what are we targeting? 7.4 in the middle. Okay, got it? Okay, so how do we measure these values in the blood? So we can go to the arteries, and one can get blood from the arteries and send it to an analyzer, and we can actually analyze the arterial blood gas and get us our pH, CO2, PaCO2, PaO2, and our bicarb. And we write it like that: pH, PaCO2, PaO2, and bicarb. Just like I've written here. And this would be considered normal. Okay? Or what I can also do is on the vein, I can draw blood on the venous side or in a vein, and on that, the CO2 will give me bicarb. Now, that's just some basics. Now, there's a question that comes up. Well, patients sometimes I take their pulse oximeter. So here's their nail, their finger, and I put this device on them. Okay? And I put this device on them, and then it gives me a value. It reads right, and it gives me a percentage saturation. That's saturation of oxygen. So is SaO2 or saturation of oxygen different from PaO2, which is partial pressure of oxygen? The answer is that is correct. Partial pressure of oxygen is what's dissolved in the plasma. So remember, we had the blood and that had oxygen dissolved, right? Now, what did we have? We have these hemoglobin pieces. We have these hemoglobin cells, the hemoglobin on the red blood cells. So remember, we talked about in the video, when the conditions are right, the oxygen actually attaches to the hemoglobin. So if I have, just come up with something, if I have two hemoglobin molecules and they're both attached, right? The saturation of oxygen is going to be 100% because that's the percentage of hemoglobin that are attached to the oxygen molecule. So partial pressure gives you how much oxygen is there in the dissolved in the plasma. Saturation tells you how many of those oxygen molecules are attached to how many hemoglobin, what percentage of hemoglobin or red blood cells. That's what that's measuring. Okay, so there's a difference between the two. One is measured in percentages, which is saturation. The other one is measured in millimeters of mercury. One is dissolved, one is attached to hemoglobin. It gives you a percentage of hemoglobin that is attached to oxygen. Okay, let's move on. This is a good way of looking at this. So you have red blood cells that are in your bloodstream. This is the arterial side. This is the venous side. Okay. Then you have your O2. You see the red dots, the O2. And if you see, the arterial side has more oxygen. And as you start to move towards the venous side, you have less oxygen. Why is that? The oxygen is getting connected to the RBC and getting dropped off to the cells. What's being picked up? CO2. So CO2 is in the blue. So you see on the arterial side we have a few CO2 and then suddenly we're building up CO2 on the venous side. Completely normal. So if you see here, you have more O2 and less CO2. On this side you have more CO2 and less O2, and that's completely normal. And then in the green you have the bicarbs. You see the bicarbs are just floating around. There's no gradient really on the arterial and venous side. Okay? So that's how it's going. And then what happens? Remember oxygen and CO2? If conditions are right, the oxygen will attach to the red blood cells and they'll become best friends as oxyhemoglobin and they would deliver that oxygen to the cell. Okay? And then if the conditions are not right, they both would separate and CO2 can get attached to the red blood cells. Okay? Now, so this is kind of what our cross-section and blood looks like. Okay? Now, remember pH. Remember high pH? So what that means less acid, right? Less acid or more base, right? Right. Now, if you have less CO2, it gives you what? Less pH. More CO2 gives you more pH. Okay? Or other way to look at it is if you have pH. So this is what now if you have bicarb. So let's say your bicarb goes up, right? Your pH goes up. Your bicarb goes down, pH goes down. Right? More Tums, more alkalemia, pH goes up. Less Tums, less alkalemia, pH goes down. Let's revisit this again. I got this wrong. So let's do this one more time. Okay. Let's say you have high CO2. High CO2 equals more acid equals less pH. Less CO2 means less acid, means more alkalemia, means higher pH. So pH is directly proportional to bicarbonate levels. Higher pH, higher CO2. Higher CO2, higher pH. Less HCO3 or less bicarb is less pH. And also it's inversely proportional to the carbon dioxide level. So high CO2 means lower pH or more acidotic. And lower CO2 means more alkalemic or higher pH, right? And that's what I'm describing here. High bicarb means high pH, low bicarb means low pH. Tums. High CO2 means more carbonic acid means less pH. Less CO2 means Less carbonic acid means more alkalosis means higher pH. Okay? So that's some basic stuff. So that's how acid and base is maintained in the body. You have cold and you have hot. So if acid is considered hot and bicarb is considered cold, there's different levels of hot and cold in the blood, and that determines one's overall pH. You see? Easy, right? Now, that's some basics of how it works in the blood. Now, what happens on the cell level? So let's take a peek at it. And why does this matter? Why am I studying this? So let's say I eat some carbohydrates, right? So carbohydrate is glucose is C6H12O6. So it goes to the cells. If there's oxygen, it gives me what? Water plus CO2 plus 6 ATP. Water plus CO2 is what? Carbonic acid, H2CO3. If there's no oxygen, I get lactic acid. So again, I get energy. I need energy or I die. Or my cells die. So I get lactic acid. So either I get carbonic acid or lactic acid. With proteins, there's amino acids with the breakdown. In fats, there's fatty acids. So what do you see common in all of this? Acid, 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 and acid. So when I am working with the nutrients, my end product is acid. So in other words, if I don't get rid of the acid, what's going to happen to my body? It's going to become acidotic very, very quickly. So I got to get rid of the acid. What's going to happen to my pH? Acid goes high, pH goes low. How does body remove acid? The biggest mechanism by which the body removes acid is to exhale CO2. This is actual removal of acid. And how does that happen? You have the carbonic acid, right? Which is H2O plus CO2. So the water remains in the body and CO2 goes to the alveoli where it's breathed out. And whatever acid is not exhaled is buffered by the metabolic side. So the respiratory side exhales CO2 and the metabolic side buffers the rest of the acid in the body so that the pH remains within the normal range, which is 7.35 to 7.45. So why? What happens if I don't keep my pH within the normal range? The proteins start to denature. It's like egg, when you break an egg and put it on a hot plate. From functional protein, it denatures and becomes delicious protein, right? Those cells denature. There's some other stuff that it does. Let's talk about what else can it contribute. So remember, if you have acid, you have too much acid in your body, your pH is on the lower side, right? I want you to hold on to that. Let's talk about what this can do, this lower pH, okay? And we'll talk about it right now. Before we do that, let's talk about, remember we looked at the blood and we looked at RBCs, and remember there were conditions under which this dissolved oxygen would become best friends, BFFs, with hemoglobin, and they would become oxyhemoglobin. They'd get married, right? It's beautiful. The oxygen attaches to the RBC, gets to the cell, and they have a divorce, right? And that's where now RBC is single, so it picks up CO2 or carbon dioxide and takes it to the lungs. Okay, so what are conditions under which, so suddenly what becomes important are conditions under which hemoglobin or red blood cells and oxygen become BFFs, right? So we'll talk about that. Now, we've already covered what's the difference between PaO2 and saturation SaO2. Remember, PaO2 is not the same as SaO2. SaO2 measures the saturation, percentage saturation of red blood cells, and PaO2 measures the partial pressure of oxygen in the blood, and it's 80 to 100 millimeters of mercury. Saturation is greater than 90%. So what causes the, the dissolved gases to become attached to the red blood cells? And this is a concept called oxy-hemoglobin dissociation curve. Ooh, that sounds fancy. The easy way to look at this is very simple. When does oxygen and hemoglobin become BFFs or best friends? And when do they divorce? And it's important because what you want in lungs is for oxygen and hemoglobin to become BFFs. Because that way, hemoglobin can drop off CO2 and connect with O2, right? And that CO2 can get exhaled so that my body is not in acidosis. I can get rid of some of that acid. On the tissue level, what you want is the hemoglobin and O2 to disassociate so that hemoglobin is now single and can pick up CO2 and take it to the lungs. Okay, that's what you want. Okay, let's look at this curve. So this curve is you have on the y-axis saturation or SaO2 of oxygen. It's the, remember the RBCs that are saturated, and then on the x-axis you have partial pressure of oxygen. And this is your normal curve. What does this tell me? It plateaus after a while. This means around 75, 80, you can increase. So whenever you have 80 millimeters mercury, after that you can give the patient more and more, you can have more and more oxygen dissolved in their blood, but the curve plateaus and it's not going to increase the percentage SaO2 of saturation of hemoglobin a whole lot, right? That's what this is trying to tell us. And then it also says there is a significant increase from 50 to 80. The increase is very dramatic when it comes from 25 to 50. Okay? But this is the curve. Now, what if I increase the temperature and increase the acid in the body? So in your tissue, because your muscles are working, it's generating energy, and it's generating, that's the source of CO2 and all the other acids, lactic acid, fatty acids. So on the tissue level, you have higher temperature and higher acid. So the curve moves towards the right. And hemoglobin's affinity for oxygen goes down. That's what you want. You want that oxygen which is attached to that hemoglobin to be given to the cell. And you want both of them to divorce. That way, CO2 can be picked up by the hemoglobin. Okay. If the curve moves to the left, okay, the hemoglobin's affinity for oxygen goes up, which is what you want in the lungs. So when you have less temperature and your acid is less, that's when the curve moves to the left. Okay. If you look at it, when the curve moves to the right, I give a partial pressure of my oxygen is 80, right? However, when I draw a little line here, right, I see my saturation is much lower. Makes sense because that RBC, that red blood cell, has now given its oxygen to the cell, so it's not going to, and it's connected with the CO2. So it's the saturation of oxygen is going to go down because the hemoglobin or the RBC red blood cell is not connected to the oxygen. That's what you want in the tissue level. And again, if you are thinking of curve moving to the left, this is where if you see with 80 partial pressure of oxygen, hemoglobin and our red blood cells and oxygen become best of friends, so the saturation is much higher, right? That's exactly what you're expecting. Okay, so the curve essentially tells us when oxygen and hemoglobin are going to be BFFs, and basically when the temperature is less and the acid is less, that's when they become best friends. When in the lungs, generally the temperature and acid is lower and they become friends. And on when it gets to the capillaries, to the cells, the acid is more and temperature is more, and it gives off the O2. And it's important because that creates space for carbon dioxide to be picked up. So that's oxyhemoglobin dissociation curve. Okay? Now, let's say systemically my pH is very, very low. Okay? That means in my lungs, the pH is going to be low as well. Because guess what? My pH is low everywhere in my body. What does that mean? That means that oxygen in my lungs, which was supposed to become best friends, right? But because there's more acid there than it's supposed to be, because I have a disease or there's an abnormality which is causing my pH to go down, that acidemia is doing what? That acidemia is preventing that hemoglobin binding to the oxygen. Ah, so in other words, let's say I have a disease and that disease makes me really sick and causes me to become acidemic, overall very low pH. Well, the pH is going to be lower in your peripheral tissues, but now it's going to be lower in your central tissues as well, which is your lungs. Which means this curve, the affinity of hemoglobin to oxygen, is going to go down. Which means in the lungs, even though I am breathing in oxygen and it's getting dissolved in my blood as PaO2, it's not going to saturate my red blood cells. You with me on this? Which means my cells are not going to get oxygen, which means what? They're going to continue to do what? Anaerobic respiration. We don't want that. that's going to continue to build up more and more acid, and proteins are going to denature. And that's why it's important to maintain pH within a certain range for the body and for the body to work correctly. Okay? Perfect. So now we know oxyhemoglobin dissociation curve and how things are going within the arterial, within the blood. Okay? Looked at it in a bit more detail. Okay, so we talked about why pH is important. Okay, so let's look at the next thing. Let's see how it all works together. So pH is body's thermostat, and that's what's targeted. Okay, and usually the processes are two kinds of processes. And this is another thing that confuses people, but I want to separate it out right now. There's a cause and there's a compensator. Okay, so the processes can either cause it or they can compensate for it. So what does that mean? Let's take a look. Let's say you have uh, let's say you have pH of 7.34, right? Let's say that's the acid you start off with, right? You want the normal pH of 7.35 to 7.45, right? So the process that causes this pH to go down is going to be the causer. Okay, the process that's going to compensate for this lower pH by increasing the pH is going to be the compensator. Okay, or the compensation. So that's one way to that's the best way to understand this. Okay, now next, lungs. So remember, respiratory side means lungs, means CO2, PaCO2. Immediate compensation. Respiratory side is fun. It starts to compensate immediately. Completes full compensation in 24 hours. Okay? How does it do it? You ventilate more. How do you ventilate more? I can do it two ways. I can increase the rate at which I'm breathing, or I can increase the depth and the volume that I'm exchanging. So respiratory rate is I breathe more number of times a minute. Tidal volume is the amount of breath I take, the volume of breath I take per breath. So if I increase my respiratory rate and or I increase my tidal volume, I increase the amount that I ventilate. If I decrease both of them or any of them, I decrease the amount that I ventilate. If I increase the tidal volume or resp rate, I'm going to abbreviate it by TD, or increase the RR, which is respiratory rate, I increase the ventilation. If I increase the ventilation, I blow off more CO2, which causes a decrease of PaCO2 in the body. Which causes what? Your PaCO2 goes down, so my acid goes down, which means my pH goes up. Okay? Vice versa. If I go down on my tidal volume and I go down on my resp rate, right? I ventilate less. I blow off less CO2, my PaCO2 goes up, my acid goes up, my pH goes down, right? And it's immediate. And there's only two ways I can change it. My respiratory side compensates immediately, changes PaCO2 using my lungs by changing tidal volume and respiratory rate. And we now know how. So we look at pH of 7.34, which is less than normal pH. So if I were to compensate, if my respiratory system was to respond to this and compensate, how would it compensate? Let's think through this. So pH is low. Okay, pH is low. So my compensator has to do what? Increase the pH. Okay, how do I increase the pH? What? Through my lungs. How would lungs or my respiratory side or my PaCO2 lead to increased pH? So remember, if PaCO2 goes down, there's less acid or there's increased pH. So my respiratory side has to decrease the PaCO2. And how does it do it? It will increase it by increasing my tidal volume or my respiratory rate. Remember, we just looked at it, or both. So in other words, if I become acidotic or acidemic actually, my respiratory side can compensate by breathing faster or breathing deeper, or both. Okay? So that's compensating. Let's flip the script. Let's say my pH is high. How would my lungs compensate? How would my respiratory system compensate? Let's think through this. My pH is high. I want to compensate. So my respiratory side has to decrease the pH. If I increase the PaCO2, I have more acid and my pH is going to be down. How do I increase my PaCO2? By decreasing my tidal volume and or respiratory rate. So again, slower breaths, lower amount of volume that I breathe in or out when I ventilate. So respiratory side holds on to more carbon dioxide. It's exactly what this talks about. Okay? That's what my body tries to do. It senses what the pH is, and then it tells to it asks the respiratory system to either breathe faster or slower, deeper or shallower, to compensate. And that's what respiratory side does. Okay? What if it's a culprit? What if it's the causer? So let's say I have asthma and my CO2 is trapped and I can't exhale it. Now here, it is actually my lung disease that is not doing a good job, and I am actually having high PaCO2 because of my disease, which is causing what? Low pH, which is contributing to my acidemia. So again, respiratory acidosis is contributing to my acidemia. So here, this is actually cause. Okay, so again, here you look at PaCO2, and if it's low, it's going to cause what to the pH? It's going to lower the pH because it's going to cause more acid. Okay, so you see the difference when the process is the culprit. When respiratory process is the culprit, the PaCO2 lowers the pH. Okay, that's what the respiratory system does, is it contributes to the pH getting worse. Here, the PaCO2 is changing to compensate or make the pH better. That's the difference. Okay? Again, if respiratory side is a culprit and you have a higher pH, what do you expect the CO2 to be? PaCO2 to be? Remember, again, low acid, low PaCO2 means low acid, meaning higher pH. So again, there's a couple of ways the questions can be asked. A, this is your pH. If the respiratory side was the cause, what would you expect the PaCO2 to be? So again, if you have low pH, I'm going to expect my PaCO2 to be, I'm going to say that if my respiratory site is causing this, then my PaCO2 is high. Okay? If my pH is high and I'm in alkalosis, alkalemia, if the cause is respiratory, what do I expect PaCO2 to be? Lower, because that means less acid means more alkalemia. The other way this question, the other way questions come up is, let's say your pH is low. Well, how would the respiratory side compensate? Well, it would compensate by increasing the pH or decreasing the PaCO2 by managing the respirate and or the tidal volume. And vice versa. So the questions can ask you, hey, how's this side going to compensate? Or, hey, if you see this pH and it was causing the bad pH, what do you expect their value to be? Okay? And if you see those values are a little different, what respiratory side does to compensate is different from what respiratory side is doing to contribute. Okay? That's very important to tease out what the question is asking you. Okay? Very good. Got it? Maybe listen to it just one more time to get a better sense of what it is. Okay? So again, if CO2 is the culprit or causer, essentially high levels of CO2 is causing high acid and low pH, carbonic acid. If CO2 is compensating or respiratory side is compensating, we are expecting it to change CO2 to get the pH in the normal range. Okay? It's very important to understand this concept because questions can ask you both those things. Kidneys. Let's go to the metabolic side. Metabolic side is governed by the kidneys and it's bicarbonate and it's the buffer. And it's a delayed reaction. It's not immediate like respiratory. It takes 3 to 5 days for full effect. And it increases or decreases bicarbonate in the blood. If it increases bicarbonate, again, if it's the cause, then increasing bicarbonate causes increase in pH and decreasing bicarbonate causes decrease in pH because decrease in bicarb means less alkalemia or more acidemia. Okay, let's say, so it's a culprit. So let's start with this being the culprit or cause. So if I see a pH of this, what am I expecting my bicarb to be if the carbon dioxide is normal? So again, bicarb will be high, which would lead to more Tums, lead to higher pH, because there's going to be less acid. If it's compensating, if it's 7.48, how do you expect the kidneys to fix it? Well, the way to fix it is to lower this pH. So the pH has to be lowered. And it's going to be lowered if the bicarb goes low. Because bicarb going low means more acid is retained. Aha. So excrete more bicarb. If the pH is low, what do you expect the kidneys to be doing? What if kidneys were the cause, what would you expect the bicarb to be? What would you expect the bicarb to be? Low. However, if the pH is low, what would kidneys do to compensate for it? Increase the pH, meaning retains bicarb or increase the level of bicarb in the blood. Okay? Now this is a little on the confusing side, so I wouldn't be surprised if you need to listen to this video again, but let's do a quick recap. Okay? So in this video, what did we talk about? We talked about how we measure in different values, pH, PaCO2, PO2, and bicarb. Okay, we looked at how it works in the blood. Why does our body need to manage the pH and oxyhemoglobin dissociation curve? Okay, and then we looked at what the body does. Okay, and what it is the OSs or the processes can cause or be the culprit of pH. So one of the ways the questions can be asked is, hey, if you see a low pH and your metabolic side is good, what do you expect your respiratory side to be doing? Or they can say, look, you have low pH. How do you expect your respiratory side to compensate? Those are two separate questions. Treat them as two separate questions. Because when the respiratory side is compensating, it's compensating to increase the pH. When the respiratory side is the culprit, it's responsible for pH becoming low. You see the difference between the two questions? Okay, it's very important. Respiratory side, you manage two things. Respiratory side is immediate, compensates immediately. It's got lungs involved. It manages PaCO2 and it manages it by the amount of ventilation of air that it does, and it does it by increase the respiratory rate and the tidal volume. Okay, and then we did a few examples and discussed what was culprit and what was the causer. And then we talked about the metabolic side with bicarbonate, which is delayed and happens in the kidney. And again, if it's the culprit, if it's causer, and then if it's a compensator, what would it do? Okay.